Hello, everyone. Thank you very much for joining us today. This is the second of our webinars for Invasive Species Week. So I'll hand over to our speaker in a minute, but I'm just going to go through a few quick housekeeping things to start with. So we're recording the webinar today, and just to make you aware, we'll share this afterwards for anyone. If you have any problems and can't hear, you can see it afterwards, and for anyone who can't attend as well. If you are having problems, you can try logging out and joining again. That seems to work sometimes, but otherwise, yeah, like I said, it'll be available afterwards, so you'll be able to catch up. And we'll have some time for questions after the talk. So if you can pop any questions in the chat box or the Q&A, we'll have a look at those at the end. But yeah, so I'm very pleased to hand over to Lori Lawson-Handley, who's going to talk to us today about the use of eDNA in detecting invasive species. So yeah, thank you very much, Lori, for joining us. And yeah, I'll hand over to you.
Thank you so much, Lucy. First of all, can you see my slides and hear me okay?
Yep, that looks perfect.
Great stuff. And well, thank you so much for inviting me to present at Invasive Species Week. It's something that I've followed for many years now, so it's really nice to be a part of it. And it's such a great way of getting the message across about invasive species. So thanks very much for the invitation. So my research has focused on invasive species for really quite a number of years now. And my main interests are trying to better monitor species using environmental DNA. So that's what I'm going to talk to you about today, our recent research that focuses primarily on Dracaenian mussels, so, the zebra and quagga mussels. So you probably don't need any introduction at all to what we mean by an invasive non-native species, but just in case you're new to this, there was a really fantastic report that was produced by IPBES last year that really sums up the issues of invasive species perfectly. So some really alarming statistics that came out from that report included that more than 37,000 invasive non-native species have been introduced by human activities globally. And we've known for quite a long time now that they're one of the main drivers of biodiversity loss globally, alongside things like land and sea use change, overexploitation, climate change, and pollution. And invasive species are linked to very dramatic changes in biodiversity and ecosystems across the world, but also they're costing us a lot of money in terms of their management and mitigation. So it's been estimated that invasive species are costing over $423 billion on an annual basis. But one of the things about invasive species, of course, is that we have a lot of control in terms of preventing and mitigating their spread if we can detect them at a very, very early stage. So early detection is really key for invasive species in order to be able to monitor and manage them effectively. And that's where environmental DNA comes in. It's been recognised now for a good few years that it's an important tool in the early detection of invasive species. So there are a number of challenges that face us when we're trying to monitor invasive species generally, not specifically with eDNA, but in general. So, you know, often it can be like we're looking for a needle in a haystack, just like Sean is here. And sometimes species can be very, very difficult to identify. They may look very similar to other species. They may be present in their juvenile stages, which makes them very, very difficult to separate. And sometimes the methods that we employ accidentally catch non-target species. So gill netting here, for example, does not discriminate between invasive species and native. So sometimes some of the survey methods we use can cause harm to the environment. So where does environmental DNA fit within our surveying toolbox, which may consist of lots of other things, for example, kit net sampling or microscopes or audio moths and camera traps and that kind of thing. So what do we actually mean by eDNA? So Wherever you're sat at the moment, you'll be surrounded by a soup of eDNA. It's present in the air, it's present in water, it's present on surfaces and in soils and every substrate you can think of. And this is DNA that's left behind by an organism as it moves through its environment. So if I, for example, touch my desk, I'm leaving DNA on the surface of the desk. As a fish swims within a lake, it's shedding cells and leaving mucus that can be detected in the water. And we can collect this environmental DNA from different substrates. And the most commonly used substrate is water that we can collect from ponds and lakes and rivers, for example. But we can also collect samples such as soil or plant material and that kind of thing. So when we collect that eDNA, it's obviously not just from a single species, it's a soup of DNA from all the organisms that are living in that sampled location, but also in the wider environment, because the DNA will run off the land and into the water, for example. So that helps to overcome that issue of looking for a needle in a haystack, because you're sampling quite a wide area. And then we can use the information from environmental DNA to either look at the presence or absence of species, which obviously is a really important thing when we're detecting invasive species. We can use it to describe whole communities, because from a single sample, we can get lots of invertebrates, we can get vertebrates and all sorts of things. And we can look at interactions between species. And ultimately, this is really useful to help us try and understand not just the presence or absence of an invasive species, but potentially their impacts on the wider community. And in order to use eDNA effectively, we need to understand a little bit about its ecology in the natural environment. So its origins include, for example, cells that have been shed by a species into their environment. It could be released by spawning events. It's influenced by biomass, so more bigger species means more DNA. And also we can get things like the physiology of organisms affecting how much they shed. So eDNA exists in the environment either within intact cells or tissues, or it can be present in organelles like mitochondria, or it can just exist as free-floating DNA without any protective structures around it. And that affects how long it sticks around in the environment. So obviously, if it's just a single molecule as opposed to being protected by tissues, it's going to persist in the environment for a lot less time. So there's been quite a lot of work done to try and understand how long DNA sticks around in the environment, and the estimates vary from approximately a day in a small pond to up to thousands of years in the permafrost. So we can detect eDNA, for example, from mammoths if we go to places in Siberia and collect permafrost samples. And that fate is very much influenced by ambient conditions in the environment. So things like temperature, UV, pH, and microbes. And finally here, we need to think about how DNA moves around in the environment. So rivers are sometimes called conveyor belts for eDNA. Rivers can transport DNA up to 10 kilometers downstream, it's thought. But also you get vertical transport in all types of water body. But I think an important thing to realize is that I referred to eDNA as this soup earlier, but it's not homogeneous soup. So maybe if you think of it as a vegetable soup that's got chunks in it, it's quite heterogeneously distributed in the landscape. And that's important because it can really help us to kind of pinpoint where particular species are. So I'm going to talk to you today about two different approaches for analysing eDNA, and we refer to those as targeted or passive approaches. So both approaches start with the same idea. So basically, we can apply these approaches to any kind of eDNA sample, whether it's water or soil, etc. I'm only going to talk about water today, though, so we'll focus on that from now. So if we're taking a water sample, we typically collect about 2 litres. So here is one of my PhD students, Sarah Peixoto. She's collecting water to look for zebra mussels in a local lake. So she's collected about 2 litres, and then she filters the water through a syringe that has a filter on the end of it, and then the DNA sticks to the filter, and we then use that DNA on the filter for any downstream analyses. So imagine you've got your filter with all the different soup of DNA in there. We can then either decide to just focus on a particular species, say, for example, our zebra mussels. And if we want to do that, we can use designed specific assays that will only target that species. Or we can use what's called a passive approach, where we basically sequence all the DNA that's in that sample and then assign the sequences later. So that gives us information about the whole community. So I'm just going to go through those approaches in a wee bit more detail, because I'm going to talk about them a bit further on. So with the targeted approach, we have 2 different options. We can either use an approach that's been around since the 1980s, which is very, very standard in all labs around the world. This is known as polymerase chain reaction, or PCR. And for this approach, we design very specific primers, short sections of DNA that target the species that we're interested in. And then we use that method to literally just look at presence or absence via a band on a gel. So if you see a nice clean band on a gel like you have here, then that tells you that your species is present in your sample. Or we can use a slightly fancier method that uses an extra level of specificity. So it's got this probe in there, which is also specific to our target sequence. And we can literally count the number of times that probe attaches to the sequence using something called quantitative real-time PCR or digital PCR. And that gives us an estimate of the number of copies of DNA that's in the sample, which we can use as a proxy for abundance. So that's a more sensitive method, and it also has the advantage of giving us information about abundance too. So the other approach, the passive approach, is often referred to as meta-barcoding. So in this case, we've got our tube of DNA with all the different species DNA represented. And then we use those primers, those short bits of DNA to stick to the DNA that's in the tube. But in this case, those primers are conserved across lots of different species. So, for example, we might be interested in targeting all invertebrates or all vertebrates. So we'll then PCR with those conserved primers and then sequence the PCR products in a method known as high throughput sequencing. And that gives us between about 50 and 100,000 sequence reads per sample, which is a lot of data to work through. We then have to use bioinformatics approaches to match the sequences to reference databases. So this is really reliant on having very good reference databases, as you can imagine. And those reference sequences are sometimes known as DNA barcodes. So once we've got that information per sample, we can look at the relative number of the sequence reads for each of the different species that we've detected. And so we've got an indication of both presence absence, and we've also got the number of sequence reads that we can use as a proxy for relative abundance. That's a wee bit controversial, and I may get some questions about that, but certainly our research has demonstrated that it gives you a pretty decent estimate. Right, so on to some key questions that you might have in terms of monitoring invasive species with the eDNA. So these are things that we've been thinking of for many years now, and that, in order to roll out these methods, these are questions that we need to really fully understand. So our first one is, how do these methods compare to established methods such as kick sampling or camera trapping or bottle trapping for amphibians or any other kind of survey methods? How do they compare for INS monitoring? Because we obviously want methods that are going to be really effective and cost effective as well. Our second question is how sensitive and reliable eDNA is, and can it give us any information on abundance? Thirdly, we need to know whether the method works equally well across all different environments. So there's a lot of interest in whether the method works in flowing waters in particular. So a lot of people kind of assume that it's much more difficult in rivers because you've got that dilution effect. And in order to put forward some recommendations, we need to know whether it matters if, where and when you sample. And then finally, ultimately, what we're aiming for is methods that are ready for operational rollout, that, that can be used by organisations like the Environment Agency and others to detect invasive species. So how do we know if these assays are reliable and ready for rollout? Well, we've got a European Union cooperation in science and technology partnership called DN to really thank for this, because they've really addressed that question very effectively. So what they came up with was a 5-level validation scale where each of those levels corresponds to a level of validation of a particular assay. So level 1 basically means that virtually nothing is known about how the assay performs. So an assay has been designed on target tissue, but it hasn't really been tested in the field. And at the complete opposite end of the scale, level 5 means that it's been through every kind of rigorous test you can imagine, to the point where we really understand how it works in the field. And I think once we get to level 5, we can really consider an assay to be operational. So for this for it to get to this level, we need to understand the limits of detection, the probability of detection, and how detection is influenced by different environmental variables. So basically, we've been working on Dreissenid mussels for a few years now to try and get to the stage where we're at level 5 on that validation scale, so that eDNA can be rolled out for routine monitoring. Now, you may have heard more about Dreissenid mussels in Invasive Species Week. They're such a high priority group of species, but just in case you haven't, so I'll just give you a short introduction to them. So Dreissenid mussels include zebra and quagga mussels, but they're both quite different species. They have very different invasion histories. So both species are from around the Pontic-Caspian area, and they've spread into Europe. The zebra mussels were first recorded in the UK in the 1820s. And as you can see from this NBN Atlas map that I pinched off the NBN Atlas this morning, they're really quite widespread in the UK. So over 4,000 records, and really quite widespread in England and Wales. Quagga mussels, by contrast, so they're from a very similar part of the world, and they've also spread to both of these species have also spread to North America. But you can see that in the UK, they have a very different invasion history to zebra mussels. So far on NBN, there's only 124 records, and those records are very much concentrated around the London area. It's a decade. I just realised it's a decade ago since quagga mussels were first detected in the UK, but certainly their invasion history is much more recent. So the reason that we're concerned about these species is because they have some very serious impacts on several different levels. So these species are what's known as ecosystem engineers. They create new 3D structures, and they increase the water transparency because of their very high rates of filter feeding. This can have knock-on consequences for the structure of benthic communities, changing their composition. And it's also pretty negative for other mussels, such as unionid mussels, because the zebra mussels in particular will use them as hard substrates, eventually suffocating and leading to mass mortality. So these bottom two pictures, you can imagine, I think, why organizations like the water industry would be really concerned about these species, because they can have, really cause a lot of damage to infrastructure by clogging and corroding pipes. And it's been estimated that these species cost about over $50 billion between 1980 and 2020 in terms of their economic consequences. So in order to effectively use environmental DNA, we need to understand the ecology of these species. So we know that with the dreissenids, their spawning is very much influenced by the water temperature. So their larvae are detected in water plankton when the temperature reaches above 12.5 degrees. And we therefore expect the environmental DNA concentrations to be highest in the summer, because we'd be picking up the larvae. We also know that these species have a preference for hard substrates, so we might expect the eDNA concentrations to be higher next to hard substrates. And we also know that, obviously, because they're mussels, they're very limited in terms of pH and calcium. So those kind of environmental variables might also influence eDNA detection. So we've had two PhD students who have both now finished working on this for the last few years. And Rosie Blackman and Sarah Peixoto. And these are some of their objectives. So basically, what we wanted to do was to design and test targeted assays for zebra and quagga mussels, aiming for level 5 on the operational scale. We wanted to test their efficacy compared to other survey methods such as kick sampling, to understand how detection varies over time and space, compare targeted versus non-targeted eDNA methods for detection. So that's basically targeted versus metabarcoding, to update the distribution of quagga mussels, and finally to provide recommendations for dreisenid monitoring going forward. So Rosie started off on this journey many years ago now. I think it was actually 2015. Started right at the beginning by designing species-specific PCR assays for both species. And these were then tested on target and non-target species and in some mesocosm experiments. And then she went out in the field to the River Raisbury for quagga mussel and the River Welland for the zebra mussel to compare how eDNA performed versus kick sampling. So basically what she found, just briefly, is that the assays are species specific. So these bands on the gel here demonstrate that the assay amplifies for the quagga mussels and the zebra mussels, but not for any non-target species. And then in the field trials, it was really quite clear that the eDNA method outperformed the kick sampling, with 100% detection in all the using eDNA for all the quagga mussel samples, and 78% detection for eDNA for zebra mussel, compared to much lower detection rates for kick sampling. So then moving on to Sarah's PhD. So what we really wanted to do here was to get these assays up to level 5 validation. So Sarah did 2 big studies, one looking at temporal variation and one looking at spatial variation in zebra mussel eDNA. So the next few slides are just going to focus on zebra mussels. So here we wanted to know how eDNA concentration varied throughout the year. When's the best time to sample? How environmental variables influence detection, and what are the limits and probability of detection? So basically, all the variables that are around this mussel here are the things that we investigated. So to do this, Sarah looked at the temporal variation in eDNA concentration in a reservoir and a river over the course of a year. And she looked at the spatial variation of eDNA concentration in 20 different water bodies in the summer. So we predicted the eDNA concentration would be higher in the summer, consistent with spawning, that it would be higher in lentic versus lotic systems and higher next to hard substrate, and that it would be positively associated with variables such as temperature and pH, etc. So to carry out this study, so Sarah took water samples, as I mentioned before, from Ekup Reservoir and the River Hull between December 2020 and November 2021. So in each month, she collected 10 samples from each of the two sites, each 2-litre water samples, and also 1-minute kick samples at each site. And we also put some colonisation plates up at Ekup, but I won't really talk about that today. The colonisation plates are to look for juveniles, I should say. And then in the second part, the spatial study, these are the 20 sites across the country. So it included 5 each of canals, rivers, lakes and reservoirs. And all these water bodies had known populations of zebra mussels. And that sampling was carried out in summer 2021. And all of these samples, and it was a lot of samples in the end, were qPCR'd with an assay that had been previously published, and we used 6 qPCR replicates per sample. So just to run through the results that we found. So the first thing that actually surprised us a bit was that eDNA was detected all year, but peaks in the summer. So this is just for the Ekup and River Hull. So obviously, as you can imagine, the water temperature peaks during the summer. So it reached 12.5 degrees, which is the critical point for spawning in April, and then peaked during June and August, as you can see in these graphs here for the River Hull on the right and Ekup Reservoir on the left. So what we were a bit surprised by was that we detected mussels all year round at both sites. So we actually had 100% detection in all samples at Ekup throughout the year. This was a bit lower at the River Hull, so we had 20% detection in January, but during the summer months, this peaked to 100%. And as we predicted, the DNA concentration peaked in the summer. So these graphs here are for the DNA concentration is on the y-axis, and then you've got the month on the x-axis. And you can see clearly that in the summer months in orangey red here, both for Ekup and the River Hull, that's when you get the peak in DNA concentration. And then on the bottom, these are heat maps that help us to understand the spatial variation within each month. So the y-axis here are the 10 different samples that we collected in each water body in each month. And you can see that there's very little variation in the columns. So the DNA concentration is quite uniform within site, but not between site. So where you have this yellowy green here, particularly for Ekup, this is the peak in DNA concentration in the summer. So little spatial variation, but obviously a huge amount of temporal variation. So the second main result from the temporal study was that temperature, not surprisingly, given what I've just told you, is the main predictor of zebra mussel eDNA concentration. In fact, it's the only significant predictor, but it was only significant for the Ekup Reservoir and not for the River Hull, even though you can still see quite a strong trend here. The other variables, quite interestingly, showed positive trends for the reservoir, but negative trends for the river. So this suggests that there may be some important differences between water bodies in terms of the eDNA dynamics. So basically, that's what we went on to study with the spatial study. So first of all, we found, again, really high detection. So 100% detection across all samples and 85 to 100% detection across replicates. So basically, if zebra mussels are present, you're going to be able to find them with this method. So this graph shows you the different types of water bodies separated out. And you can see that reservoir here in red has got slightly higher DNA concentration compared to the other 3 water body types. And that difference is significant, just about. There's no significant difference between the other types of water bodies, though. And then the second key result here was that the eDNA concentration was slightly higher next to gravel. So here we've got sand, silt, gravel, and boulders on this graph here. But there's no significant difference between boulders, sand, or silt. So to be honest, I don't really think that the substrate makes a huge amount of difference here. And then the final important thing from the spatial study was that we found a significant effect of volume filtered. So when we filter the water, we record how much we can pass through the syringe, because sometimes the filter clogs and it can be difficult to get all the water through. So these models tell us that that there is a significant effect of volume. So on this graph on the left-hand side, we've got some of the key environmental variables that we were testing. And if these lines overlap the red dotted line, it means that there's no significant effect. So you can see that the only one that does is volume. But actually, even though that's significant, when we looked at detection probabilities, so we did some modeling to try and understand how volume impacted the probability of detection, we found that it was very, very high, even if you just have 2 samples and you filter over 750 mils. So basically you have a 99% detection probability, even if you just filter 750 mils, which is pretty good going. So taking all this together, just to kind of summarize these key results. So first of all, we've got that temperature is the key determinant. So if you sample in the summer, you'll maximize detection. So going forward, a really important recommendation is to sample in the summer so that you really maximize your detection. The second one is that detection is slightly higher in reservoirs compared to other water bodies and slightly higher closer to gravel. But I don't think that this is too much of a concern, because the detection and the concentration of DNA was generally really quite high. And then finally, we found a small but significant effect of the amount of volume of water filtered, but you can achieve a 99% detection probability with just 2 750 ml samples. Now, although we found this, I would never recommend that you only take 2 samples because of the needle in a haystack issue. And particularly if you're looking at sites where you don't know how well established the mussels are. So always take a minimum of 10 samples, but with this method, and if they're very established populations, then in theory you should only need 2. And we'd conclude from this that at least the zebra mussel assay is ready for operational rollout. So I want to move on now to just focus on quagga mussels. And we started working on the Raspberry system a good few years ago now. And the first thing we did was to compare targeted methods versus passive methods versus kick sampling for detecting eDNA of quagga mussels in that system. And to do that, so Rosie went out and she collected 3 500 ml water samples at 6 points along the Raspberry River. So you can see this rectangle here is Raspberry Reservoir, and then you've got the river coming out here. So these diamonds are the points that we sampled along the river downstream. And what we found was that detection of quagga mussels was 100% for both the qPCR and the regular PCR assay. It was a little bit less for metabarcoding, but eDNA was generally much higher than for kick sampling. And what we also found was that there was a gradient in the DNA concentration with both qPCR and metabarcoding. So as you go further downstream, you have a dilution effect, but you also probably have fewer mussels going from the reservoir downstream. So this concentration of DNA, the concentration of mussels, is reflected in the concentration of DNA. So we've recently gone on, well, fairly recently gone on to tweak that assay to make it even better. And then we've tested it in 24 new sites across the summers of 2021 and 2022. And I have to say, unfortunately, the results are pretty depressing because we've managed to confirm 11 new positive sites for quagga mussels. So if you recall earlier, I showed you a map with the NBN distribution. That's basically these black triangles on the map here. The circles that are on this map are sites that we included in this current study. The squares are ones that we'd included in our previous study, and the is it hexagon here is one that David Aldridge had sampled back in 2023. So where you've got an orange circle, it means it's been tested, but quagga mussels weren't detected. Any colour of blue means that they were detected, and quite alarmingly, they're now up as far as Nottingham and Loughborough. In water bodies such as the Grand Union Canal. So I know anyone that's has a concern about freshwater invasive species starts to panic when we know that they've got into canal systems, because we don't want a repeat of what happened with demon shrimp. So unfortunately, we do have a positive detection of them in the Grand Union Canal and also in several rivers. So we've got them in the Cam, the Nene, the Little Ouse, the Great Ouse, as well as the Thames and Wraysbury, which we already knew about. The other thing is they're also in very high activity lakes and reservoirs, including Willen Lake, Ely Marina, Rutland Water and Holmpean Point, which all have very high water sports activity. So this is a real concern, and it's really a call to arms to implement some biosecurity at breakneck speed. So just looking finally at the DNA concentrations for each of those samples. So on this graph here, so on the left-hand side, you've got the DNA concentrations from our previous study, and then on the right, you've got the ones from the current study. And I think it's quite informative to look at the differences in the DNA concentrations in the different water bodies. So where we've got really established quagga mussel populations, the DNA concentration is high, as you'd expect. We've got some populations such as Bottisham and Ely and various places here where the concentrations are very low, and we might stand a really good chance of getting in there early and preventing any further spread where there's low concentrations. So you might consider that those locations are really high priority for management, or at least effective management. So finally, just to finish off with some summaries and recommendations. So we demonstrated that eDNA outperforms kick sampling for detection of both quagga and zebra mussels. I'm pretty confident that the methods are sensitive and reliable enough, and also provide some information on abundance. Detection rates are high across all environments. They're a bit higher in reservoirs, but you certainly don't need to be put off by how they perform in rivers and canals. They work absolutely fine. We can detect them all year round when we've got established populations at least. But if you want to maximise the chance of picking them up, then sampling in the summer is the right time to do it. We've demonstrated that a targeted approach is more sensitive, but something I haven't talked about today is that one of the advantages of metabarcoding is that you're not just surveying one species, you're finding out information on the whole community. And twice now we've detected new non-native species using this approach, which I wish I had time to talk about today, but I don't. So we'd say that certainly the zebra mussel assay is ready for rollout. And to be honest, given the high priority with the quagga mussels, I say it's at level 4, but it's good enough, and just go for it. Especially if it's you kind of an early warning tool, then I would certainly be confident about going out and using it in anger. And finally, just a bit of a sobering one to finish off, further monitoring and swift action is needed to try and slow the spread of quagga mussels. So that's me done. Thank you very much for listening. There's been a lot of people that have contributed to this, a big collaboration over the years, but mainly Rosie Blackman and Sarah Peixoto carried out the work as part of their PhDs. And we've had funding from the Environment Agency, Yorkshire Water, NERC, Anglian Water, the FBA and CH. If anyone wants to know more about the quagga mussels, you can click on this QR code. The paper is out on, it's open access on bioRxiv. So that's it. Thank you very much for listening.
Brilliant, thank you, Lori. That was excellent, and this work is really useful to us as well. So we're doing some work at the moment to improve biosecurity at quagga mussel sites. So we're going to include your sites in that.
Great. I need to send you some more detail on that.
No, that would be really good, really useful. I've seen, so we've got a few minutes for questions. I've seen one come through already. So
stop sharing my screen then.
Oh yeah, that's a good idea. Does the size, like the wide size variability of invertebrates, have an impact on the amount of shedding of eDNA particles, and can that be accounted for in analysis?
That's a very good and very difficult question. So we try to investigate that with our mesocosm experiments. So actually, at the same time as we were running some mesocosm experiments with the quagga and zebra mussels, we were also doing the same kind of thing with killer and demon shrimp. So the first obvious thing we found was that not surprisingly, really, the mussels shed a lot more DNA, produce a lot more DNA than the shrimp do. So first thing, it's going to be a lot easier to detect a mussel than it is to detect a shrimp. So yeah, so certainly the form and function of your species is going to have a big influence. I think because of the filter feeding biology of mussels, you'll always have a good chance of detecting them. They just produce loads of DNA. We did also just do some experiments where we put the different numbers and different weights of those species in the mesocosms, and we did find an effect of abundance or biomass during the DNA accumulation part, but then it kind of it stabilized. I think you get this kind of like complicated feedback loop of this interaction between production of DNA and degradation of DNA, and then it all gets a bit horribly complicated. But yeah, certainly I think you can have at least some proxy for the number of mussels that are present in a water body, even though it's rough.
Thank you, Lori. Another one here. So higher water temperature could also accelerate eDNA degradation and thus reduce detection. Is there any indication in your studies of an upper temperature limit where this could offset the increased shredding?
That's a really good question. And no, there isn't. And we, I don't know if you remember, was it the year before last? We had this horrendous heat wave. The reservoirs. So we've sampled at Ecup Reservoir so many times. I mean, like really many times without seeing a zebra mussel. And then we went during the heat wave, and the water level was so low that there was just hundreds of thousands of zebra mussels everywhere. And that, even maybe if you're in the south of Europe, or where water temperature gets really high, maybe there might be some feedback, some sort of trade-off, but we never experienced anything like that in the UK.
Okay. One more here. So thinking about eDNA in rivers, I have more concern about the travel distances than the dilution. How do you determine where the DNA originates from other than upstream?
That's a great question as well. So there's many experiments been done to look at traces in water bodies, and the field is really now using quite sophisticated hydrological models to really try and understand the unique characteristics of not just a particular river, but a particular part of a river, in space and time. So at the moment, I would say the estimates are fairly crude. I mean, we can say, for example, if you look at the Raspberry system, you get this dilution effect. We could give you an idea of they're no more than a couple of kilometers upstream. It's crude, but the way the field is going, there's some groups really developing some great hydrological models for eDNA. So I think that that's something that will really improve over the next year or two.
Yeah, that's great. I think that's all of the questions for now. Oh, no, one more. There's lots of thank yous as well, Lori, by the way, for a great talk. So because eDNA can be moved in water flow and introduced from outside of the system and can't discriminate between live and dead specimens, can you comment on how we can be certain a positive eDNA detection actually reflects the positive detection of a live animal?
That's a really great question as well. I think it's about having a really good sampling design. So I'd be really wary of anyone that tells you that you can, just go and take a single water sample and that'll tell you everything. I think if it's so important when we're talking about invasive species that we have real confidence in the results. So I think taking as many samples as you can afford to and feasibly process in a given area will help. Will give you a much better indication of whether something is actually live or dead. And also, if you have a positive result, then go back and test it again. I think so DNA degrades quite quickly, so I think that it's unlikely that you'll get a really continuous signal if you've just got a single if you've got a dead mussel and someone throws it in a pond, you're going to be able to detect it for a few days, but then that DNA will degrade and it won't persist over time. So good spatial and good temporal sampling is the key, I think.
Yeah, great. Okay, I think that is. Oh no. So would eDNA water sampling be effective at detecting terrestrial species at low densities present in the water catchment?
That's great. That's really good questions. So I've always been a bit more comfortable, with my eDNA hat on, thinking about water. But we've recently been working quite a lot on terrestrial rewilding projects, and we've got a pretty good. It's pretty good for detecting animals that are in the wider catchment. Certainly so most of this we've done on vertebrates. I think it may be harder for invertebrates, but because there's lots of runoff and animals come to water bodies to drink and interact with the water. So you do get that runoff into a water body. So I think that's really useful because it may be that you're interested in detecting, I don't know, let's take something really obvious like a grey squirrel. You wouldn't use eDNA to detect a grey squirrel, but something obvious like that. You will be able to pick them up in the water body, and that will give you an indication of if they're in the wider landscape. And if that's the case, you can then design some more rigorous surveys to look at different spatial scales and things. But yeah, I think it can give you a pretty good indication of what's in the broader catchment.
Yeah. Okay. Right. I think we probably are going to have to finish it there. But if any other questions do come through, I will pass those on and we can share any answers. Someone's asked about the presentation. So we'll make this recording available afterwards, and then there's the QR code in there, isn't there, where people can get that paper as well.
Absolutely. My email address is in there as well. If anyone's got any particular questions, I'm more than happy to chat.
Yeah. Brilliant. But yeah, thank you very much, Laurie. That was brilliant. And yeah, another great talk for the week.
Thank you so much, everyone. Thank you for all your great questions, and yeah, I hope everyone gets a lot out of the rest of the week. I'm sure it's going to be great.
Yeah. Thanks very much, everyone. Thank you. Bye.
Bye.